Walter Isaacson. Walter, it's so good to see you. Thank you so much for doing this.
Oh man, Katie, and thank you for doing it. So great to be with you again.
Well, you write so many fascinating books about so many fascinating people. To name a few, Leonardo da Vinci, Ben Franklin, Steve Jobs, Henry Kissinger, men all, I might add, although I'm sure there's some women in your Innovators book,
Mm.
Walter. But tell us about what inspired you to write your next book, to really roll up your sleeves and dig into a woman named Dr. Jennifer Doudna.
I was interested in the next great innovation revolution. We've had sort of the physics revolution at the beginning of the 20th century, then the information technology revolution in the second half of the 20th century, and now, because of CRISPR and gene editing and now COVID and vaccines, we're entering a new era, which is a life sciences revolution. The molecules will be the new microchips, and our kids will not only have to learn binary code, but genetic code. And the person who best represents this is Jennifer Doudna because she figured out the structure of RNA back in the 1990s and then helped develop CRISPR as a tool to edit genes. And now we're using messenger RNA to do vaccines. And she was inspired by reading the double helix and reading about Rosalind Franklin, who was the great scientist who came up with some of the pictures of DNA, but she never got the credit that James Watson and Francis Crick got on
Right.
DNA. And she said, okay, I want to be a scientist. And her high school guidance counselor said, women don't become scientists. And so she did. So I wanted to inspire people that we should all love the beauty of nature. And by the way, that women can win the Nobel Prize like Jennifer Doudna did this year.
Yeah, we'll talk about the Nobel Prize in a moment, but I'm fascinated about her, by her upbringing because I think it really informs especially little girls in terms of what they believe they can become. She faced obstacles, but what was it in her personality, Walter, that really prompted her to persevere? And what about the things that shaped her externally as well?
First of all, she was curious, just like Leonardo da Vinci to Steve Jobs. That's a key trait of anybody I've written about. And her curiosity growing up in Hawaii was about things like the sleeping grass. You'd touch it and it would curl. And you and I have seen things like that that happen in nature. But she just kept obsessing over, well, how does it do that? What makes it curve? And all of these things in nature that she found miraculous. And so she wanted to figure out the underlying molecules in nature and how that would work. Secondly, she was persistent and competitive. She loved playing soccer growing up in Hawaii. She was a bit of an outsider because she was, from born of the mainland and was blonde and lanky. And so they called her names in the small school she was in Hawaii, a Haiole, which is not a nice name, but so she felt like an outsider and that also helped. But I think being a woman and figuring out that she was being left out of the sequencing of DNA, the Human Genome Project. So a group of women in my book, Jillian Banfield, Emmanuel Charpentier, Jennifer Doudna, they focus on RNA. So those are the traits growing up, being a bit of an outsider, being persistent, and being passionately, obsessively, and even playfully curious.
Well, let's talk about her discoveries. As you mentioned, she, along with Dr. Emmanuel Charpentier, I hope I pronounced that correctly, Walter, won the Nobel Prize for Chemistry for CRISPR. Before we get into RNA and mRNA, messenger RNA, can you explain what CRISPR is for a fifth grader? Because you're so good at breaking things down. And we've been hearing about CRISPR, Walter, for a few years now, but I still think people scratch their head when they hear the acronym.
It is both a simple and a beautiful concept. Bacteria have been fighting viruses for about 1 billion years longer than we have, and they developed a system which has clustered repeated sequences in their DNA. That's where the word CRISPR comes from. And those clustered repeated sequences contain some of the genetic material for any virus that has attacked them in the past. So that virus attacks again, it knows how to chop it up. It's an adaptive immune system, just what we need these days. And so what CRISPR does is it can target a sequence of DNA, and it uses a scissors, an enzyme, but it's like a molecular scissors to cut the DNA right where it's targeted. And what Jennifer Doudna and others did said, hey, we can turn that into a tool to edit our own genes. So it's pretty simple. It's just a molecular tool that's a scissors that'll cut DNA in the spot that the RNA tells it to target.
But there are a
Is that
lot of
simple
people
enough?
who
Did that
worry.
work?
Not really, but Walter, we'll talk offline and you can give me an even simpler explanation. But, I know a lot of people are really worried about this technology. They think it's incredibly promising, which clearly it is. We've seen that in COVID, which I'll talk to you about in a moment. But there are a lot of fears about CRISPR, about designer babies, about mad scientists. Talk about the pros and cons of this exciting discovery.
What it is able to do is edit in embryos, edit human embryos, so that you have inheritable gene edits. In other words, we can edit our species so that our kids and all of our descendants will have different traits. That sounds like sci-fi, but in 2018, just 3 years ago, a Chinese rogue scientist who had been to Jennifer Doudna's conferences was able to edit in as embryos two twin girls, so they didn't have the receptor for HIV, the virus that the receptor, the virus that causes AIDS. And you think, well, that's a great thing. They'll never catch AIDS. But it also caused a lot of, shock and awe because it means you could edit, certainly things like Huntington's disease, sickle cell anemia, that are simple genetic fixes. That's probably good. But soon you'll be able to edit for muscle mass, which is pretty easy, or height, or skin color, hair color, eye color, and even memory and processing power and IQ. So there's a lot of problems with that, one of which is that the rich will be able to buy better genes for their kids. You'll be able to go into a clinic and say, okay, I want my kid to be taller and more muscular and this, that, and the other. And that's a problem. And secondly, it will harm the diversity of our species. So one reason I wrote this book is that it's a wonderful miracle thing that'll help us fight things like COVID, but also sickle cell anemia. But we got to be careful. I mean, this is like Prometheus snatching fire from the gods. We are able to hack our own genes, and no species has ever been able to do that before. So we need to figure it out.
That's scary as hell, I think. You think of like designing a super race or, what does this mean? And are there, in fact, are people thinking about this ethical dilemma? Have any guardrails been put in place, or is this just something that people are talking about in the theoretical sense?
You talk about a super race. Jennifer Doudna one night after she invented this CRISPR gene editing technology had a nightmare in which she's brought into a room to meet somebody who wants to understand it. And the person looks up and it's Adolf Hitler wearing a pig face. And so she decides, okay, we're going to have to gather scientists from around the world and figure out what are the guardrails? How are we going to try to stop it? Which is hard to do because even I was able to go to a lab with two of Jennifer's graduate students and edit human genes using CRISPR. Now, don't worry, we flushed it down the sink with some chlorine, but we have to be sure we can keep this genie in the bottle. Now, we're going to want to use it to fight things like viruses. We are going to want to use it to fight things like Huntington's and sickle cell and cystic fibrosis. But I think we have to have rules that say you can't use it for anything that's not medically necessary, and you can use it to fix diseases, but you can't use it to enhance our children, because that's where I think you get into danger. But there's been a lot of discussion about this, and the ethics of it is very much a part of the book where it's complicated. We got to figure out what do we want to do.
Well, how do you, I guess the question is, Walter, how do you establish and enforce those rules? Who makes the rules? And, can there be assurances that those rules won't be broken? Honestly, it does have the makings of a really terrifying Black Mirror episode.
Yeah, I mean, you could have medical tourism too if the United States decides to ban it, but somebody in an offshore island decides to have a fertility clinic where they edit babies for you and let you design your own baby. However, we are able to find international rules of what we're doing. You know, Peggy Hamburg used to be the FDA commissioner. She's one of the co-chairs of
Right.
an international commission that's trying to set the rules. And even China, with this rogue scientist edited the babies 3 years ago, has now totally banned it. So you'll never be able to stop it totally. You can't stop, trafficking in ivory tusks or shoplifting, but I think we can try to control it if we have the will to do so. But also, we got to be careful we don't control it too much because we're talking, you and I are now talking about, well, that's kind of bad to be able to design our children. But there's some good things too. We want to be able to design our children so they, maybe that they don't have Huntington's or Tay-Sachs or sickle cell or cystic fibrosis. And maybe even we want to design our children for other good traits. So I
Well,
think we've got to be
yeah.
careful before we knee jerk say, hey, we don't want to ever fix genetic problems.
Definitely. And what about things like cancer? Obviously there's a strong, I mean, they're not always hereditary, but they can be, and there can be a strong predisposition, knowing what I know about colon cancer and pancreatic cancer, which killed my sister. Do they think there's a lot of promise in the world of cancer as well?
Absolutely. And you know a whole lot about colon cancer. Also, unfortunately, I'm sorry about pancreatic cancer. And it's not just the heredity component. You can use CRISPR to be able to detect cancerous cells and then actually, as part of an immunotherapy system, try to attack them. So these are the many, many good things. Even now, both in China and in the United States, we're starting to fight cancer in clinical trials using CRISPR.
Wait, can I ask you a dumb question? Because Walter, it seems to, how do they actually do it? How do you edit and splice genes? Because I'm trying to picture it and I can't quite do that in my head.
Well, there are different ways. Suppose you want to fix sickle cell, which they've done this past year and cured a woman of sickle cell in Nashville, Tennessee. They take out the blood and the marrow, the cells, and you edit the cells, and you can put them back in the body. That's easy to do with blood cells. You can do it with the eye. They're trying to fix, some forms of blindness by doing that. The harder one, although it'll be cheaper and easier in the long run, is to take an early stage embryo or a sperm or an egg, and then you can edit that in a petri dish easily. You can say, okay, we're going to take out the gene for Huntington's, we're going to put in a gene for more muscle mass, or take out the gene that stops muscle mass from growing. And then you do that and you implant it, in an in vitro fertilization clinic.
It's really amazing, isn't it? When you think of the technology, it's mind blowing. And of course, now with COVID, I mean, talk about perfect timing. Unfortunately, we're not
Mhm.
celebrating, obviously, this horrific pandemic, but it really is quite relevant, her research, to coming up with the vaccines, which are using messenger RNA. Can you explain how her discovery has been implemented in the production of vaccines?
Yes, about a year ago in March when the pandemic first struck, Jennifer Doudna has a 17-year-old son named Andy. It packed him off on the train to Fresno, California in order to be in one of those robot building competitions. Middle of the night, she wakes up her husband and says, we got to go drive and pick him up. I've been hearing about this pandemic. I don't want him to be in this big conference center in a robot building competition.
Right.
Of course,
A
the
super
kid is a
spreader
bit
event. Yeah.
right. The kid's a little bit upset, but they drag him away. And just as they're leaving, they get a text message saying, okay, robot competition canceled because of COVID. And that's when Jennifer decided she was going to turn her attention to scientists fighting COVID. Now, you can use CRISPR as an easy detection technology to just spot the genetic material of the coronavirus. So that's being rolled out now, which is CRISPR detection tech. You also can use CRISPR to do what bacteria did, to say, I've spotted this virus, the genetic material of it, and now I'm going to cut it up and I'm going to kill it. And another thing is I just got to yesterday my second Pfizer vaccine. That, as you said, is an mRNA vaccine, just like Moderna. That uses RNA as a messenger to tell the outer part of our cells where proteins are built, which proteins to build.
Epigenetics? The epigenetics,
No,
is that
no,
what it
yeah, no,
does?
it's a cytoplasm, which is the manufacturing region of a cell. And so our
I was trying
DNA
to
is the
sound
most
smart, Walter. Dude, I was trying to
Epigenetics
sound
is really
smart.
cool and really smart, and I wish I could weave it in, but I'm not gonna try. DNA is the famous sibling. It's the one that gets put on magazine covers. But like a lot of famous siblings, it doesn't do much work. It just sits in the nucleus of our cells curating information. But the RNA goes there, copies some of the information, goes to the outer region of the cell, and builds proteins. So what an RNA vaccine does is it said, all right, here's the code to build the spike protein, a part of the spike protein from a coronavirus. So it builds a fake coronavirus spike protein that goes into my blood system, and my immune system kicks in and says, we don't like that spike protein. That means if I ever get hit by the real, coronavirus, it'll kill it because my immune system has been stimulated to do it. All of this comes from the use of the most miracle molecule, which is RNA. And Jennifer, as I said, in the 1990s, did for RNA sort of what Rosalind Franklin did for DNA. She helped figure out the structure of it, as did Jim Watson and Francis Crick, the double helix. And figuring out how RNA works will be the most important thing we're going to do to fight cancer, to fight viruses, to edit our own genes, because RNA is the thing that tells our cells what to build.
So has she been intimately involved, Walter, with the development of these vaccines? And with Dr. Fauci saying we're entering an era, sadly, of pandemics, I imagine she is going to be absolutely instrumental as perhaps more variants, I hate to even say it out loud, show up, or even new viruses.
I think a lot of what she is working on, and the people who are sort of her rivals, the guys at the Broad Institute at MIT and Harvard, working on the next generation of things. In other words, right now, my arm is killing me. My wife is, I got a headache because our immune system has been stimulated to fight the virus. If you could make it work just like CRISPR, which is to say, instead of stimulating my immune system, we are just going to attack the virus directly. There's something called Carver, which is coming out that Feng Zhang and others have worked on at MIT. There's something called Pac-Man. They all have wonderful names that comes out of Jennifer Doudna's orbit, which will, in the future, we'll be able to code it and say, okay, this new virus is attacking us. It'll take 20 minutes maybe to figure out the sequence and say, we're going to code this scissors to chop up that virus. So that's going to happen, and detection technology is going to happen. And that will solve our Dr. Fauci crisis of new pandemics coming along real quickly.
But how long does that take? Isn't there a process where they have to identify and figure out and all that? I mean, how quickly can this technology respond to new challenges?
Very quickly. As you saw with Moderna and Pfizer, it really took them less than a month to take the sequence of the coronavirus and then build RNA that would fake a spike protein for it. In some ways, once the sequence changed, once the virus mutates, whatever, we're talking about a day or two really to find the RNA or the scissors-like system to kill it. What's hard is how to figure it out, how to put it in these lipid nanoparticles so they can put in a syringe and get it into my body. And that's why you have to keep them cold. It's the delivery system that's the harder part, making the new things that'll either chop up the virus or stimulate our immune system. That'll be done pretty quickly every time we get a new wave of viruses. Heck, that's what bacteria have been doing for 1 billion years, and that's what CRISPR teaches.
Yeah.
That's how what Jennifer figured out is that CRISPR teaches us how to do it.
What about collaboration? I know that are they working together? Is she working together with the MIT scientists? Because obviously, when you're talking about the survival of the human race, you would think that people would put their competitive natures aside and work together. That is happening, isn't it, Walter?
Yeah, and this is the end of my book, The Code Breaker, which gives it a wonderful ending, I think, which is she and Emmanuel Charpentier were in a terrible competition, strong competition with the people at the Broad Institute of MIT and Harvard. And this wonderful scientist there named Feng Zhang, born in China, raised in Iowa, a really sweet, good scientist. They both came up with ways to use CRISPR, which had been, discovered by Jennifer Doudna and Emmanuel Charpentier. But then it took them another 6 months to come up with ways to use it in a human cell in order to edit human cells. And that was in early 2013, and they're still in a patent battle over who has the patents to use it. That'll eventually be solved. But what was really cool is in March, when both the people at the Broad Institute and then Jennifer Doudna's group out in Berkeley and in the Bay Area of California first started doing things like detection technologies for the coronavirus, ways to kill the coronavirus. Instead of trying to patent it or own the intellectual property, they instantly put it up online night after night. Each of the labs would be putting things online so it'd be in the public domain. And that helped kind of restore the sense of the scientists are doing it because it's a noble pursuit.
That's awesome. Walter, I marvel at your ability to jump into these really interesting areas of study, whether it's computers with Steve Jobs or foreign policy with Henry Kissinger, the world of art, science, and invention with Leonardo da Vinci, and Ben Franklin, obviously, and everything that he accomplished. I mean, you must be such a quick study. How do you learn and learn the vocabulary and learn to speak the language of all these brilliant people so quickly?
Well, especially with the Code Breaker, this new book, there's a joy in figuring out how something works. And that joy is especially great when that something is ourselves. So I love it. I think it's important on all of us. If we're humanists, I think it's really bad to say, I can't understand science. It's bad for the scientists to say, I don't like poetry, I don't like art. Because it's Steve Jobs over and over again insisted to me when I was working with him. It's the people who can connect the humanities to the science, who stand in both realms that are the Leonardo da Vinci's. That's what Vitruvian Man, that guy standing in the circle in the square, a self-portrait of Leonardo. It has a spiritual quality because people who love both the humanities and sciences tend to be the most creative persons. So I think, to me, being curious about all aspects of nature, that's what distinguished Leonardo. He loved anatomy and he loved art. He loved zoology and he loved music and he loved math. Likewise, at Jennifer Doudna, I mean, the reason I love this working on this book is that she's a brilliant scientist, but she also cares about women in science, and she also cares about the ethics, about the morals. And so that type of connection has always excited me. And look, Katie, you have a long career of being interested in all sorts of things. It's fun to be interested in all sorts of things. It's fun to be somebody like yourself.
Well, I think curiosity, as you said, which really motivated her from a very early age, so important to stay curious for your entire life. And by the way, I just want to say, so important to demystify science for people, because this is when science people are starting to not trust science as much, not appreciate scientists as they should. So this is a real love letter, I think, to scientists everywhere. And that's so important, especially, I think, in this day and age for obvious reasons, which we could have a whole nother conversation
You're right. I
about
really
truth
want this to be a
decay and
love
the,
letter. Yeah. I really,
yeah, and the
you're so
book
right.
is
I
called,
want it to be a love letter.
it's called The Code Breaker, Jennifer Doudna, CRISPR, and the Future of the Human Race. Walter, it's always so fun to talk to you. Thank you so much.
Thank you, Katie. I get energized every time I talk to you.